I will be talking today about disparity among the patient population in clinical trials in neuro-oncology. The literature is mostly based out of the United States, but certainly some of this information is applicable to Europe and other parts of the world as well. So here are my disclosures. So let's first off by explaining what disparity means. As per the Merriam-Webster dictionary, it's a noticeable and usually significant difference or dissimilarity. So why do we care about disparities in clinical trial populations? Well, when trial populations don't mirror the incidence of disease, the reproducibility and generalizability of results are limited. And if there are biological differences between populations that could influence efficacy and or toxicity outcomes, then the clinical trial data cannot be extrapolated from one population to another. So meaning if you do a study only in men, can we really extrapolate that information to apply to women as well? When a more diverse population participates in clinical trials, we increase the potential to know more about the extent to which different subgroups might respond to a medical product. So what kind of disparities might exist in clinical trial populations? Age, gender or sex, race, ethnicity, socioeconomic disparities, sometimes leading, say, to less access to care. Populations with special health needs, such as comorbid medical conditions or disabilities, and geographic differences. Now, we're going to focus mostly on the top three topics for today. So first, speaking about age, as you all may know, older patients are often excluded from trials. And in this figure, you can see the global age standardized incidence rates per 100,000 populations of CNS cancers by age and by sex, with the pink color representing males and the blue color representing females. And you can see that CNS tumors in general occur at a higher incidence rate in older populations, but yet they're often excluded from clinical trials. And just as an example, as we all know, the median age of diagnosis of glioblastoma is around 65. But if you look at the pivotal trial, which demonstrated the benefit of adding temozolomide to radiation, the median age in that population was 56 to 57. And this is an issue widespread in oncology trials. An analysis of phase 3 randomized clinical trials demonstrated that the median age of trial participants was a mean of 6.5 years younger than the population-based disease site-specific median age. Now, how are older patients excluded from clinical trials? Sometimes there are eligibility criteria that strictly restrict based on age. Sometimes they're excluded indirectly, either by having too high of a functional status or demanding good organ function and limiting comorbidities, which often can be seen in older populations, such as cardiac disease or renal dysfunction. What about the in-between, the adolescent and young adult population, ages 15 to 39? Again, this figure demonstrates the distribution of AYA cancers across the age, and you can see that CNS tumors in this light green section constitutes a decent proportion of the AYA cancers that we see. AYA cancers represent 6.8% of all cancer cases worldwide. And even though we have seen improvements in the survival of AYA patients with cancer, those improvements in survival still lag behind their pediatric and older adult counterparts. When studies have been looked at to see why this is, one of the possibilities that's speculated is perhaps the barriers to clinical trials for AYA patients. But when it's been actually examined in studies, the enrollment rates of AYA patients in clinical trials vary, with some studies saying that there are limitations and some studies saying that there aren't as many barriers as we suspect. That being said, as you can imagine, traditionally, pediatric trials have enrolled patients up to the age of 18, while adult trials have enrolled patients no younger than 18. And so what do we do about a patient, say, who has a pediatric type tumor but is age 21, or has an adult type tumor but is age 17? Those patients wouldn't necessarily be able to enroll on the trials if we have those strict cutoffs. And we have to think about this particularly with respect to the new WHO 2021 guidelines, which now describe pediatric type versus adult type diffuse gliomas. eEANO and the European Society of Pediatric Oncology released a guideline advocating for AYA access to clinical trials that was published earlier this year. And the FDA also provides guidance on when to allow patients as young as 12 to participate in adult cancer trials. So we do have to actively think about how we should design trials, keeping the AYA population in mind when appropriate. Next, I wanted to talk about gender and sex, which is often used interchangeably, but they do mean slightly different things. So does gender or sex impact disease? Well, there are studies that demonstrate that women have a 1.5 to 1.7-fold greater risk of developing an adverse reaction to a drug. And within neuro-oncology, we know there are studies that in newly diagnosed glioblastoma, that female sex is a predictor for severe myelotoxicity from temozolomide. Their studies also suggest that sex contributes to differences in cancer risk according to tumor types, even after controlling for known epidemiological risk factors, and that sex contributes to differences in treatment responses. So this slide is not to bog you down with a lot of details, but there have been a number of regulatory policies that have been enacted specifically regarding women in trials. And again, I apologize, this is mostly speaking about the FDA. Through an unfortunate history of several drugs, which unfortunately led to consequences in women. For example, thalidomide given to pregnant women, resulting in birth defects. In 1977, the US Food and Drug Administration released a guideline advising the exclusion of women of childbearing potential from early dose ranging studies. And even though they didn't mean to exclude women in general, this was ultimately misinterpreted to meaning to exclude women in general, such that the NIH had to establish a policy almost a decade later encouraging the inclusion of women in studies. But when the US General Accountability Office then examined the state of the literature in clinical trials, it was noted that women were still being routinely excluded from clinical trials when those trials were supported by federal funds. So this led to further enactment of additional policies, with the FDA releasing a guideline reversing that 1977 guideline and endorsing the representative inclusion of women in phase 1 to 3 studies. In 2001, again, the General Accountability Office looked at the state of federally funded clinical trials. And even though the number of women being included in clinical trials was increasing, women at that time were still only 22% of participants in the initial small-scale safety trials, which has implications, as I mentioned, if women perhaps may be more at risk for toxicity. These early safety trials really look at that toxicity. In 2005, the EMA also concluded that women were slightly underrepresented in phase 1 and 2 trials, but not phase 3. And there have been a number of regulations now really calling for a balance of sex in not just human studies, but also even in animal studies. A number of funding agencies have also called for increased transparency when reporting about the role of sex as a biological barrier. And ESMO in 2018 provided consensus recommendations for studying sex differences in studies. So with all these regulations in place, do gender disparities still exist in clinical trials? And this was a study that was just published earlier this year in Cancer, looking at gender representation in clinical trials, leading to FDA approval of oncology therapeutics between 2014 and 2019. And even though the sex differences in clinical trials varied by cancer type, the authors still felt that women were more often underrepresented compared to men. This is a comparison of the male and female oncology clinical trial enrollment in orange compared to US cancer incidence in blue from 2014 to 2018. And you can see that females are still slightly underrepresented. Now, you can also look at the enrollment incidence disparity, which represents the difference between male and female trial enrollment compared to the national incidence. And depending on the cancer, right, sometimes males are overrepresented, sometimes females are overrepresented. But if you look at sort of the major cancers, colon and rectal cancer, still females are underrepresented. If you look at lung and bronchus cancers, females are underrepresented. Kidney cancer and renal pelvis cancers, women are underrepresented. What about specifically in phase 1 trials? And the studies looking at oncology trials in the literature currently represent small numbers, so I decided to instead talk about the study which looked across the board at multiple indications, not just oncology indications. This was also published earlier this year, an aggregate analysis of clinicaltrials.gov. For all studies that were registered between 2000 and 2020. In the end, it included over 20,000 interventional studies enrolling approximately 5.11 million participants. And you can see, depending on the phase of the trial, the representation of females with those in later phase clinical trials. There's an equal representation of females versus males, but in earlier phase 1 trials, females are still slightly underrepresented compared to males. So what about race and ethnicity? And I should pause that this is a very complicated topic in the United States, and I'm sure as well in Europe and other parts of the world. And there was an elegant perspective piece that was published earlier this year in the New England Journal. And I'll just read their first line from that article, that in the United States, race, ancestry, genetics, and medicine are inextricably linked in a complex and fraught history. So why do we study race? Well, race is directly associated with genetic ancestry and therefore indirectly related to genetic variants that may affect disease and health outcomes. But race and ethnicity represent more than just that. Race and ethnicity are self-ascribed or socially ascribed identities, and they can influence socioeconomic position. And even within a single race or ethnicity, there is substantial variation. So we certainly have to realize these points when we're looking at race and ethnicity. But can race and ethnicity impact disease? Again, because they're indirectly related to genetic and epigenetic variants. There are examples in the literature of this. I've just provided a copy here with the first genetic variant at the 6q25 locus has identified Latina women as being associated with protection against breast cancer, although this originates from the indigenous American population. So it's not routinely true across Latina women. It's those who have this indigenous American ancestry. And here's an example of carbamazepine and the increased risk of skin toxicities, these particular skin toxicities in Southeast Asian populations due to the higher prevalence of this HLA allele. So before I talk about race, I do want to clarify what I mean by race. And again, because most of the literature comes from the United States, that's why I'm providing these categories as defined by the United States. Of course, every country is going to have their own set of mix of different ancestries, so may or may not always be applicable to your specific population. But the United States identifies 5 racial categories: white, Black or African American, American Indian or Alaskan Native, which refers to the original peoples of both North and South America, as well as Central America, Asian, and Native Hawaiian or other Pacific Islanders. And then there's a separate ethnic category, Hispanic or Latino, which refers to people coming from Spanish culture or origin, regardless of race. So the FDA is heavily invested in promoting the collection of race and ethnicity data in clinical trials. In 2014, the FDA released an FDA action plan to enhance the collection and availability of demographic subgroup data, and their guidance document was updated in 2016, really to improve the completeness and quality of demographic subgroup data collection, to identify barriers to subgroup enrollment in clinical trials, and to make this data more transparent. And so because of this, we have now these FDA drug trial snapshots. And here's just an example from the 2020 drugs trial snapshot, which looks at the drugs that were approved in that calendar year. And you can see for each of the drugs, the percentage of women that enrolled in the trials that led to that FDA approval, the race ethnicity breakdown, as well as even how many of the patients were age 65 and older, and whether those studies were run out of the United States or outside of the United States. And so just as an example, this is a drug, avapritinib, which is FDA approved now for GIST with PDGFR alpha mutations. And you can see there's even a section that asks the question, were there any differences in how well the drug worked in clinical trials among sex, race, and age? So we can take that information and then compare it to the incidence prevalence in the population. And here comes from an interesting article that was published by ProRepublica, looking at ALK-positive non-small cell lung cancer. Now, the prevalence of ALK positivity within the Black community in the United States is similar compared to whites as just in general the populations for non-small cell lung cancer. So you can see that Black patients have a higher number of new cases of non-small cell lung cancer is higher among Blacks than their white counterparts. But if you look at the clinical trials that led to the FDA approval of brigatinib and alectinib, which are both drugs, ALK inhibitors, you can see the severe underrepresentation of Black patients. Similarly, if we look at prostate cancer, which is a disease that tends to affect Black patients more than white patients, and also the risk of death is higher in Black patients than in white patients. You can also see that FDA approval of this prostate cancer drug, the severe underrepresentation of Black patients in those clinical trials that led to FDA approval. So what do we know specifically about disparities in neuro-oncology clinical trials? Well, there's less literature about this, but we're going to go through the literature that is available. So Yang Liu and the team from University of Rochester looked at disparities in patient enrollment on glioblastoma clinical trials. They included studies in the United States that had been completed, enrolling adult patients, interventional studies, and all phases 1, 2, and 3. And there were 118 trials that were eligible for analysis. And what they found is that in general, the interventional trials in glioblastoma significantly underrepresented older patients. So you can see that the median age over these timeframes ranged, right, 52 to almost 60. But again, the median age of diagnosis, depending on which study you look at, is somewhere in the mid-60s. That older patients were less likely to enroll in early phase and non-randomized trial. The authors concluded that although fewer women were enrolled in glioblastoma clinical trials, they felt that their numbers generally reflected the population. You can see it down here, the percentage of women that participated in glioblastoma clinical trials, somewhere around 37%. In general, the breakdown of women who develop glioblastoma is around 43%. So numerically lower, but they didn't feel that it necessarily reached statistical significance. They also tried to look at racial disparities, but there really was insufficient data about these subgroup categories to be able to comment on the role of race and ethnicity. And in their conclusions, they felt that clinical trials should be designed with eligibility criteria to be inclusive of older patients and their comorbidities, and that clinical trial data should be published even if negative and should report data on race. So this group also tried to look at the role of race and ethnicity, and they also looked at brain tumor trials on clinicaltrials.gov, but they did not restrict necessarily just to glioblastoma. And what they found really is that race is often not reported or underreported, with only 28.4% of the literature publishing the demographics of participants. But in those studies that did include demographics about race, they concluded that whites were significantly overrepresented in trials for metastatic brain tumors and high-grade glioma trials, that Blacks, African Americans, Hispanics, and Asians were significantly underrepresented in high-grade trials, and that Blacks and African Americans were underrepresented in trials for metastatic brain tumors. So as was previously mentioned, we've been leading a collaborative effort between the Society for Neuro-Oncology, eEANO, patient advocacy groups, clinical trial cooperative groups, and other partners to really try to increase clinical trial accrual. I just include here the 2 papers that we've published this far, talking about the barriers to accrual and enrollment in brain tumor trials. There is a section on diversity that was led by Ugo Chikweke and Sean Harvey-Jumper that I refer you to. We've also published a paper trying to optimize eligibility criteria in clinical trial conduct to enhance clinical trial participation. But just to go through briefly some of the barriers to clinical trial participation, there's limited awareness of clinical trial opportunities, and when there is a clinical trial opportunity, it's often not near the patient. And there's a study that came out of University of California, San Francisco, demonstrating that minority patients with glioma who enrolled in clinical trials lived on average 30 miles closer to the cancer center when compared to those who did not enroll in the clinical trial. And that really speaks to the study burden and inconvenience. Sometimes there's overly stringent eligibility criteria, as I mentioned, particularly with the elderly population. With respect to underrepresented minorities, there's often distrust in the medical community, and there can be language discordance, as well as culturally incongruent care that could be barriers to clinical trial participation. So what can we do to reduce these disparities in clinical trials? Well, we need to know what representative means for your particular disease and set intentional recruitment goals. And sometimes for underrepresented populations, that means having to go beyond the traditional academic medical centers to access patients, whether that be community networks, smaller hospitals, direct-to-consumer campaigns, patient advocacy groups. Also, decentralized or hybrid trials may be able to increase trial access for certain populations. So in summary, age disparities exist in clinical trials, including in glioblastoma. That representation of women in clinical trials are improving, but may still be underrepresented in earlier phase trials, which, as I mentioned, can have serious implications given the possible sex differences in toxicity and pharmacokinetics. Race and ethnicity information is now mandated by the FDA in NIH-sponsored studies, which does help increase the quality and transparency of demographic subgroup data in clinical trials. And reducing disparities in clinical trials requires a concerted effort from multiple stakeholders. So with that, I thank you for your attention.